a lot needs to be invested. We've tried to do this in our institute, and this was an activity mainly driven by Felix, whom many of you will know. So Felix was on the team as a postdoc when we started the bat work in Ghana. He's now independent in the institute. He's still a member of the institute that I'm chairing. And so he's collaborated with the GIZ, or with the German Ministry of Collaboration, to bring PCR testing to many, many countries, including also on the African continent. And there is a lot to be learned, and there are really important messages here. For instance, this is an activity where the team sent out diagnostic tests, so test kits, PCR test kits, and asked for results back. So if they wanted something to be confirmed, or if a sequence, this also then involved sequencing, was to be resequenced, they offered to do this and collected the information. And of course, this brings information that you can then use for retrospective analysis. And for instance, this one is the result, the core result of a paper that will appear this week in Science, shows how, in fact, the Omicron variant that caused travel bans to and from South Africa and other African countries in December 2021 actually was already establishing and defining itself genetically from August in West Africa. This is mainly samples from Benin, but also here in Ghana, some of the samples had this virus. So had people known, had South Africa or other African countries known about this, no travel bans would have been issued. And this lost alone South Africa within 2 or 3 months about 60-70 million US dollars, just in terms of restricted travel and tourism industry. So it's not only for individual health, it's really for the bigger purpose of the population to have an overview about what's going on in the population, and there is a lot of work ahead. So for the practical things, let's switch to more the background. So let's speak a little bit about reservoirs and sources of pathogens. I've just discussed with Ben here about the possibility of looking for Marburg virus, including in Ghana. So this is the reservoir of Marburg virus, Rosettus aegyptiacus, a fruit bat, which is roosting in caves, and where you have mining, you have interaction with Rosettus and possible transmission of Marburg virus to humans. This is really under-investigated, but I think it will gain a lot of more attention now. Also, there is, in the whole field of arboviruses, a very complex ecology, much more complicated than for a mammal like a bat in a cave, and other mammals like humans just catching a virus. Here, a virus has at least two different hosts to which it has to adapt in parallel. And these adaptation processes really restrict the virus in a certain functional niche, and it's not as easy for such a virus to find a new host. Another example of the basics for pandemic preparedness research is plain ecology. In every ecology textbook, if you buy one, you will discover the first chapter always reads diversity, biological diversity. So let's look at diversity. These are studies published in the recent 5-6 years, looking at different host systems and just sequencing what you can find, right? So now with the arrival of next generation sequencing, we can sequence everything. And you see the GWAC program, it's intended to be not the last, so that's maybe a good starting point for a series. And we will discuss today the field of pandemic preparedness research. There are many notions here in this field. It's a very complex field of research, and what I will try to do is give you a perspective on this, which is certainly my perspective. There are others, and I always try to somehow provide a a systematic introduction first, and then also into what we have in terms of research problems, and then come back to, let's say, a resume. But to start, so the claim here is that we prepare for the next pandemic based on research. What do we know? We know that the next pandemic agent is likely to be again a virus, so probably a virus of zoonotic origin. This will certainly be in the sequence respiratory, enteric, or vector-borne transmission. So we'll have one of those transmission patterns. Transmission patterns are important when you consider pandemics. And we know that there is clear scientific evidence for respiratory pandemics in the past by viruses other than influenza. So I don't have to even mention influenza. This is the classical pandemic respiratory virus, but there are others. I will touch upon some of them. We also have some ideas about how pandemics come about. So this, for instance, would be a summary of something we could call the chain of emergence principle. So we are starting with a virus in an animal reservoir. We are seeing the first human case, probably without noticing it. Then we'll have a limited outbreak that's optimally already recorded and subject to surveillance programs. And then if we don't stop it, we end up in a pandemic. And we could divide this into two parts. The green one here, this somehow reaches up to the first spillovers into individual humans in single cases seen at hospitals. This is preparedness research. The rest of it actually is response. We could also make a division here based on, let's say, responsibilities. So the green field would be in the hands of researchers, while the orange is more in the hands of public health people, public health professionals. But of course, there are transition zones here in the responsibilities. There is maybe another depiction of the field, which is more like the disease ecology perspective, where you have two stages of viral emergence that are somehow, intermingled a little bit, but we can make a separation here. First barrier would be against a virus leaving the original reservoir host and infecting first human cases. You could say this could be something like a cellular barrier, tropism barrier. Then the other is an adaptation barrier. This is in fact more interesting to evolutionary biology people than the first barrier, because this is about adaptive evolution, right? Adaptive evolution is what we can somehow enlighten by concepts from evolutionary biology. So we have basic research disciplines, and they are all involved in this business of pandemic preparedness research. So the chief disciplines here are virology, ecology, and evolutionary biology. And they spell preparedness research in a slightly different way. So virologists think about viral diversity in reservoirs, not only genetic diversity, but also the functional diversity. So we can sequence the viruses, but what does it tell us, right? Then the ecologists, they think about, let's say, hosts as a situation that a pathogen is in, like a situation that an animal would be in. So you could equate the host with the habitat. This is an interesting concept and provides a lot of food for thought and concepts for research. So we could ask for how frequently do pathogens spill over. So this is like the frequency of habitat change, and then conditions favoring spillover and what happens genetically during this process. And then in proper evolutionary virology, as I said, we're looking for adaptive changes, adaptation. Sometimes surprising things happen. Sometimes what happens in early transmission chains is not adaptation, but neutral evolution. So virus diversity, and I'm trying to become a little bit more practical in the beginning of the talk, not in the end, but in the beginning. What do we know? This, in fact, is very basic virology, stamp collecting, characterization of pathogen diversity. So this is a slide summarizing studies that went on around the globe about describing bat diversity, bat virus diversity targeted to coronaviruses after the emergence of the first SARS coronavirus in 2002, 2003. Kumasi was one of the places where this took part, so we had a whole series of projects with a team here in Kumasi characterizing this type of virus diversity. If we compare what we knew before SARS in terms of the coronavirus tree, this was not a densely populated tree. This was really like a very thin tree in winter. 10 years after SARS, there are many more branches known, and the more branches we know, the better we can orient ourselves in the genetic diversity and then, for instance, come up with diagnostic assays. So here you see how we can populate the tree over time. There is SARS, there is the MERS virus, then comes SARS-2, which is very closely related to SARS-1, nothing new, not a big challenge anymore. And this puts us into the position of, well, providing a diagnostic test very fast, within less than a month, having a diagnostic formulation not only, let's say, conceived, but also validated in terms of this has been tried and tested on real clinical material and it's proven, it can be used in labs all over the globe. We started in Germany to distribute this, but was very soon after adapted in many other countries. And in Germany, for instance, this put us in a position to discover our first wave in the spring of 2020 based on lab testing, rather than based on intensive care units overflowing like in Italy. And this probably saved Germany about 60,000 lives in this spring 2020. We have a very old population age profile, and the population is quite vulnerable against such a virus. Quite a direct effect of laboratory testing. In countries like Ghana, big parts of Africa, we have another issue when we're speaking about laboratory testing, which is capacity building, right? So here in Ghana, you now have quite some capacity established after the pandemic, and also before the pandemic, there was quite something going on. So you were in a position to do PCR testing. This is not the case for most regions in most countries in Africa. It uses less cytokines, different types. There is less cytokine induction, and then also the resilience against interference. So if we have interferon around and the cells become somewhat reluctant to support virus replication, something we call the antiviral state that a cell assumes in presence of interferon, we can see that this new virus is much more robust against the effectivity, the effects of interferon. So there is something going on here about a change in virus-host interaction. We can't really pinpoint it now. We have further studies going on to find the molecular reason for this and to also map this difference in the genome of viruses, because there are changes all over the genome and probably only a few of them explain this phenotype. But a phenotype, to know there is a difference, it's not just a different sequence, just a combination of letters, but there is a different behavior of the virus. This tells us a lot. So we can say that this MERS lineage 5, circulating since 2015 in the Middle East and having outcompeted the other viruses in camels. So humans are only taking samples from the camels, but something happened in the camels, and we can see the effects of this still in human cells. We can test the effects in human cells in interferon experiments that we know are relevant for virus host switching or host acquisition or whatever you want to call it. So it seems viruses can be selected for these effects in their natural reservoir, and this selection also still affects what happens or what can happen in human cells. This is what I, as a virologist, would refer to as let's say, a more valid or useful insight that goes beyond sequencing and that may approach the concept of pandemic preparedness research, not surveillance. So surveillance is the key for the practical response. For those who like advanced phylogenetic trees here, so with some of the trees in this picture have a larger genetic diversity than cellular life. right? So this is viral diversity. It's vast genetic space. And every red line here is something discovered anew in just one study, just by sequencing. So this blows your mind. This, for instance, is something we did. It's on negative strand viruses in insects. Everything red is a new virus. It's crazy. And we can, by now, derive simple conclusions from this. You can just tone it down. I can put it closer here. You can derive simple conclusions that are, well, sometimes a bit simplistic. For instance, we can say there is coevolution if you consider the very deep lineages in phylogenetic trees of viruses and of hosts. You can say yes, you can overlap these trees. But as you look at units of genetic diversity that may be more medically relevant, for instance, a family of viruses. This, for instance, is paraluxoviruses. Now we refer to this not only as one family, but when we did this study, this was still considered a family. It's a contiguous tree of viruses that are all related to each other and their host associations. And as we take this apart, we can say that in this situation where we have a mammal and a virus that's typical for mammals, there is no co-segregation anymore. So there must be a lot of host switching going on here. So studies of deep diversity, so for instance, if we look at insect viruses and their deep host lineages, this will actually rarely identify new or pre-emerging human pathogens. What you see is what's around, but this is not all dangerous. That's the important message here. If you look at mammalian viruses, and you observe where actually in this system of viruses do we have atypical host associations? Where do we have typical host associations, where the diversity is much deeper? And where the chances to catch a virus from a source are much broader because there is more diversity around, well, here this starts to make sense. But what we sometimes discuss as in pandemic preparedness research actually does not even, well, address these more basic parts, but people tend to work on this aspect, on spillover. So the question, how can you catch the virus and how frequent is this? So also we try to do work like this here in Kumasi. This is the Eidolon Helvum colony in the zoo. Most of you will know that the zoological gardens here in Kumasi hold a wild colony of Eidolons that fly in each year in autumn, and they breed over winter. They leave in May to more southern parts of the African continent. These are between 300 and 400,000 animals, and you can study these animals for viruses. So we did this in a series of projects here, and well, I'm just showing you one tiny example. This is a virus we termed Kumasi raptorvirus. This grows in cell culture, and as we have the virus, we can do simple serological tests. And for instance, if we look back at the same fruit bats, about 50% of them are seropositive. If we look at swine, and these swine do not have any direct contact, but we know they eat the same fruits sometimes falling from the trees, 30% of them have antibodies. At the same time, there is no antibodies in goats, sheep, and cattle roosting in the same region. So this seems to be a specific thing. It seems to be there is a specific susceptibility of swine, right? So this becomes interesting. And then also humans. If we look at humans, two little cohorts. There is exposed humans, 45 of them, people working in the zoo. 11% have antibodies. These are confirmed by neutralization tests, so these are real antibodies. And from people from Komfanochi Hospital, non-exposed people, almost, well, hardly ever you find antibodies in these people. So also here, this seems to be a specific association. And then the literature is full of other things. For instance, this one, coronavirus, found in Guangdong province, southern China, from a bat, an alpha coronavirus, not typically known to switch hosts and infect other livestock, but in this case causing epidemic diarrhea in piglets, and this is the more worrisome thing, being transmitted between farms, right? So they really carry this from farm to farm, and they see young piglets die. Adult piglets seem to be quite robust against the disease. And it's just an observation. We can't tell how often this takes part. So, okay, something seems to be wrong here. I've lost the picture. So, without showing the pictures, I can just continue and challenge your imagination. So, so we don't need a slide. Just read this question and think about it. Do you think we can determine the frequency of viral spillover? Can you conceive this? Can anybody conceive how this could be done? In the vast geography, not even in the city of Kumasi could this be done. It's absolutely impossible. But this is a prevalent claim in the field of pandemic preparedness research, or now One Health sometimes, right? This is, I think we shouldn't make claims like this and try to sell this to politicians, right? This can't work. It's really hopeless. We could do other things. So we could say, can a better understanding of the functional diversity of reservoir-borne viruses predict their propensity for spillover? I provide you one example. This has been well discussed in the past few years, which is the existence of a furin cleavage site in the spike protein of SARS coronavirus 2, right? So is this a sign for the virus to have been genetically engineered? Is this occurring in nature? What's going on here? So revisiting a collection of rhinolophus samples containing coronaviruses, in this case from Europe, finds you, even in a rather small collection, two different viruses. This one here, one of them here in red. I don't know, can I indicate this? Yeah, this one from Slovenia, this one from Bulgaria. They have this constellation. They need one nucleotide change to develop a polybasic cleavage site. Just one change. This is happening all over the place. So with studies like this, you can answer questions that I just described. Another example, the MERS virus. This is certainly, let's say, a pandemic candidate because it's occurring in a livestock species, in camels. This is a prevalent livestock in the Middle East region. And also here, what do the sequences tell us? In fact, they tell us very little. If we had surveillance, we would be enlightened. But actually all we can do is look at repositories of sequences. So this is like the sequences you can count in GenBank that researchers put in by year from the region. And what we see is a peculiar observation. There is this red virus variant, lineage 5, taking over. So the other lineages somehow disappear by 2016. And then from this point onwards, after obviously a peak in counting sequences, we see, well, this virus somehow has outcompeted the other lineages. So if we take virus isolates, and this now is really in the center of virologists' interest to study viruses, it becomes interesting, right? So if we take viruses from the endemic region and we sort them according to phylogenetic clades, and the middle one here, the lineage 5, this is the new one, and we ask ourselves what's going on about this virus, we can do experiments. So let's look at this. On the left, we have vero cells. They do not have an intact interference system, and the interference system is the first barrier against virus replication in any mammalian cell system. So this red virus replicates to the same level as shown here on the y-axis as all the other viruses. These are the parental viruses. So this, in fact, is a recombinant virus lineage, but recombination happens all over the place in viruses, especially plastron viruses. And this is a involved virus, let's say a prototype virus from MERS. They all replicate to the same level. We take interferon competent cells and we see the red one now takes off. Another type of cell culture, another type of cell culture. So there seems to be something going on. This red virus replicates better. Also, if we use human airway epithelial cultures or human lung explants or even airway epithelium from camels, this is very hard to get. That's why there is so little replication in this experiment. We see this virus has an increased fitness, an increased propensity to replicate. Also looking at lung cuts here in these immunofluorescence pictures, it seems the virus spreads somewhat more. And we can then ask through the viral replication cycles. For instance, is it entry? Does it enter better? And here we see there is no difference in entry. This is virus pseudotype systems where you take the surface protein on a different virus and you count how many viruses enter the cells. No difference. Again, entry experiments. And then it's about cytokine induction. And here we see the virus replicates better. And at the same, so the next pandemic is likely to be caused by a virus. This is likely to be of zoonotic origin, yes. Respiratory, enteric, and vector-borne transmission, these are the transmission patterns we should look at. And there is clear scientific evidence for respiratory pandemics in the past by viruses other than influenza. I've shown you some examples. But I have to somehow amend this. So we should say evolutionary biology is fascinating, but it's unlikely to prevent or predict anything. This is really an unfulfillable promise. Spillover happens, but it cannot be measured or quantified. There are probably two scenarios for early detection when we want to become practical now. One is livestock, so we can't access all the bats on the globe. But a country may be able to assess all their cattle herds. This can become reality, right? So livestock surveillance is probably the only feasible way to do reservoir surveillance. And then, of course, humans ourselves. And here is the biggest challenge. Most countries in Africa do not have any disease surveillance in place. So it's much more likely the latter that will solve and predict or prevent, or I don't know what, hinder the next pandemic. So outbreak response hinges on detection and detection capacities everywhere on the globe. Had we seen in West Africa that something like Omicron is coming about, something could have been done, and detrimental interventions that were useless at the time they were concluded could have been avoided. And this all comes back to capacity, training, and education. Without universities in place, without training, without African scientists staying at home or returning home after being trained for a few years abroad, this can't happen. So what you're doing here is really key to pandemic preparedness and prevention. And it's the most important thing to do. So with this, I thank you for your attention. Interesting example, which is the early emergence of SARS coronavirus, the first one, the one that didn't start a pandemic. So we know these are the hosts for the virus. This is well established. But after the virus left these hosts, we also know about the sequences of the early human viruses in China. This has all been sequenced and reconstructed. And you see this bracket here saying early. These are genomes of early humans infected with SARS-1 in China, and they have a full genome. And from this point onward, the virus lost a gene. So there was a deletion of 29 nucleotides in open reading frame number 8. So this disrupts the gene, cuts it into two halves, and these halves are not functional anymore. And the prevailing assumptions this time was actually that this must be a plus for the virus, right? The virus has changed a gene, deleted a gene that must have been useful only in the host, and the virus can get rid of it in humans because it just consumes ATP in replication, and as the virus doesn't use it, it gets rid of it. This is wrong. Something else happened. So if you reconstruct this gene in a reverse genetic system, so a system where you can manipulate the virus genome and change it, and you put it back to how it originally was in the first human cases, what you see is there is a loss of function associated with this deletion. And this function can be re-established if you re-establish the gene. And this function is replication. So there is quite a striking replication phenotype here. This is the deleted virus, and this is the re-established virus. In different cell lines, the re-established virus replicates better. This is completely unrelated to effects from interferon. You can put interferon on the cells or not. The difference between deleted and full virus is always the same. And you can even take bat cells where the virus originally comes from and test the same viruses, you find the same effects. So with the full gene, the virus replicates better. So what has happened here? This is a simple case for evolutionary biologists. This is a case of neutral evolution, a loss of function, slightly deleterious mutation, Muller's ratchet phenomenon, however you want to call it. It's a founder effect, right? So we have a flock of chicken on the main landmass, and a breeding pair comes to an island, and this breeding pair happens to be less fertile. So the hen, it doesn't lay 8 eggs, but only 4. So nature has two choices now. This chicken flock cannot establish on the island, so it's extinguished because the fertility is not sufficient, or it will thrive. And we have a new species with time on this island that is somewhat less fertile, but still can exist because there is no competition, right? So this is what biologists call founder effect. In the world of viruses, it's easily expressed with slightly different words. You have a virus coming from a host, establishing an early human-to-human transmission chain, but not a raging epidemic. So this means, again, there is in the new habitat, the new host, humans, there is no competition. There's just this one virus lineage spreading, and nature can choose whether the virus is extinct, which happens in many, many cases. This is the normal case. This is the main reason why we don't always see epidemics and pandemics as soon as we touch a bat, right? People are infected. Remember the example of Kumasi raptor-virus. 11% of the zoo workers have antibodies. Still, this virus is unknown outside Kumasi. The reason here is extinction caused by founder effects and neutral evolution, because the likelihood of a mutations happen all over the place, to be harmful to a virus are much higher than the likelihoods of a mutation to be beneficial. And only sexuality in animals is what corrects this situation where we have detriment here, but also a likely benefit here and a likely benefit here. And in every generation, we bring the best of the generations together by recombination or sexuality. Viruses don't do this. Viruses are asexual organisms, and they underlie a phenomenon known to evolutionary biologists as Muller's ratchet phenomenon. So a ratchet that decreases and decreases and decreases the fitness of an organism. And this translated to transmission chains, well, it can become a very simple example. Look at this. These are all the cases of SARS-1 in Singapore. Can you see how let's say, the irregularity of the spreading pattern causes a situation where you can only just interrupt the backbone of this transmission chain here. If you interrupt one of these transmission events, this is the end of the pandemic. This is how early viruses, just after emergence, are always on the brink of extinction. And this is why we don't always see epidemics and pandemics. And this message, I guess, is much different from the message sent nowadays in the One Health, pandemic preparedness, and I don't know what you want to suggest to politicians, research terminology.